You know, was, they move it down and just say, okay, it's the temperature. It's not, that's nothing wrong with that.
Yeah, I guess you just have to go on to that second one.
We have to share the screen.
It was.
Make that
We might do
Yeah.
I mean, okay, we probably.
So this is on you.
I'm
gonna start.
Okay.
There's a PowerPoint clicker down here. If he wants to. There's a PowerPoint on the trail on the side there, and it should work.
Yeah. Yeah.
Should.
I'm like, what should have been doing wrong? But I understand that
That's operating better, you know.
not everybody has a microscope. You can take a picture of 1 millisecond. Right? And in 3D. Right? It needs to be done in 3D because it's a sheet of 3D down. Okay, this is-
Oh, there's a little thing right there.
There are lots of time marking. People want to do. That this would be. Okay, sure. So, yeah, I don't know. Yeah, I thought he was working with Joanne. Yeah.
No, we are seeing some of the alignments. I was stuff is going yeah,
Yeah, I'm not very safe. It's always a little more.
well, you're going to be
Yeah.
Very good.
Okay.
It's actually, I never know how long this. Something else.
Okay.
Important.
Yeah, I know, it's very slow.
I know how it goes.
So it's my partner and I, and it's about 11:50 total. So I'm very happy that she has a full-time job.
You should get your mic down. It's 38. That's wrong.
There's 10,000 per 15.
Sidewalk.
So,
Switches on. Don't forget your screen. That's the part. Right. It's my great pleasure to welcome Fang Huang, one of our alumnus from this department, back here to give a colloquium. Fang got his BS degree in physics from the University of Science and Technology in China in 2004. And he came here as a graduate student in the physics degree program. And he actually worked with a few of us. So his first paper here was with Diane and Jim Thomas. And I was lucky enough to then steal him away into my lab, and where he worked on analysis methods and graduated in 2011. 2011, he went to Yale as a postdoc to the lab of Jörg Fabersdorf, and he was there until 2015, when he joined Purdue University in the biomedical engineering department. And he now recently has become the Riley Associate Professor of Biomedical Engineering. He's going to give us a really great talk today. And well, I won't take away any of the surprises, so I'll just let you take over. Welcome.
Thank you so much, Keith. You hear me? Okay. Thank you so much, Keith, for the introduction.
This is slide advancing.
Good. Okay, it's my great pleasure here. So I have been spending my time at UNM, and from 2005 to 2011. And I worked in the old building of physics department, and we call it Panda. And for I think more than 5 years of research in both Dr. James Thomas lab and Professor Keith Leakey's lab.
It's fading in the outside.
Very no problem. Is that better now?
Yeah.
Okay. All right, so
Control on the white thing.
over here.
On here or on the? Right here.
Hello, hello, can you hear me? Okay. All right. Thank you so much. So without further ado, let's talk about what we, so I want to talk about today, researchers from my lab that work on ultra high resolution structure and molecular imaging of cells and tissues.
Put it on your slide.
Okay. Well, human makes observations with very different scales. For example, the galaxy that may be 1 light year, 1 million light years in diameter, all the way down to atomic level and even electrons that are 10,000 times smaller. We use different tools to make these observations, and we have to create them such that we can understand this world. Biomedical engineering now focus on, for example, fruit flies and mouse, or even full body scans of humans trying to understand disease mechanisms. Also, we want to look at cells and bacteria and viruses. In many cases, also proteins and even small molecules down to 1 nanometer level. We use different modality to do these imaging tasks. For example, we can use MRI, CT, and PET to make observations from full body human scan all the way down to potentially 25 micron level with the pixel size of an MRI scan. And we can also use electron microscopy to look at single cells all the way down to angstrom level. So light microscopy is a very powerful tool for biology and biomedical research. Due to its contrast, you can look at individual proteins with very high contrast at the same time in live cells. So it has been a primary tool for biomedical studies in life science studies. However, its diffraction limit is limited, while due to the diffraction of light, its resolution is limited by around 200 nanometers. And this actually is discovered by Abby Ernst in the last century, 200 years ago, and basically state this: structures smaller than delta R cannot be resolved. And you can see over here the definition, lambda divided by 2NA, where the lambda is your emission wavelength of your light, and NA is the numerical aperture of your objective. And that's usually around 0.2 to 1.7, for example. And this number is around 250 nanometers, and anything below this limit cannot be resolved. So this actually is carved in stone in University of Vienna, and it's basically a fundamental barrier of light microscopy that cannot be overpassed conventionally. So let's put this into a cell, how these diffraction limits affect our observation into a cell. Over here, you can see nucleus, you can see Golgi complex over here. In the plasmid reticulum, mitochondria, actin filaments, and all of these are extremely small features, and these intricate features actually governs their function. If you put a diffraction limited focus spot of, for example, good quality laser with the highest numerical aperture, what you will see the side of focus roughly around the mitochondria. That means the mitochondria, although it's very complex in its structure, it cannot be resolved. The inner membrane cannot be resolved. What it looks like in light microscope is basically a blob similar to your PSF. And we know that there's proteins that are smaller than mitochondria. For example, these transmembrane proteins and this lipid bilayers over here with a 10 nanometer scale bar on this size. If you consider this 300 nanometer point spread function over here, if I plot that, that will be basically as big as this wall. So imagine every protein emit a point spread function as big as this wall. How can you resolve the structure showing on this picture? This is another picture of showing the diffraction limit, how they can show different structure in the cells, but still the details cannot be shown. For example, over here, this is a NOT98 proteins labeled in COT7 cells. These are nuclear pores dated on the nucleus envelope. They're important for transporting RNAs. Over here, you can see individual puncta structures at different locations, indicating maybe there's an increased concentration of these proteins at certain areas. With our technology, we call it super resolution microscopy, that you can see this puncta are much better resolved and much better than the diffraction limited region. In fact, that you can zoom in for current pictures. A couple of puncta structure is diffraction limited that look like this in conventional microscopy. If you do super resolution microscopy, you can see this individual puncta are well resolved. And in fact, that they're no longer puncta anymore. You can see they potentially showing a 40 to 60 nanometer ring-like structure, indicating the epitope position of these antibodies formed in the inner channel of these nuclear pore complexes potentially form these ring structures. Comparing to other diffraction limit image and high resolution image, you can see a drastic dip increase of your resolution achievable in visualizing these cell structures. So how do we achieve that? If you look at this simulation, which actually makes this movie during my PhD, and you can see on the side is 200 nanometer, very small features. And if you put this into a confocal microscope, no matter how much dollar you spend on it, there is a hard limit of physics. That means all the structures that are on the edges that can be much better resolved than in the middle. And this basically in the middle is under diffraction limit. You cannot see the underlying structure within. So the concept is using single molecule localization. So one way or another, you only activate and allow emission of certain molecules at different frames. Therefore, at different frame, you're imaging only a subset of molecules. However, these molecules are big, but their centers can be precisely localized. And then their center position can be put in here for high resolution reconstruction. The more precise centers you localize, the better image you will get. And therefore, gradually, you can grow in this super resolution image to resolve the structure that cannot be resolved previously. And this technique has been invented in 2006 and won the Nobel Prize in 2014. Now, maybe you can imagine we can take any data and dynamic processes, potentially with unlimited resolution as we need it. And testing a biological hypothesis will be simply go ahead and take a picture or movie of the process you're trying to study with the resolution that is sufficient. However, this is not the case, and at least yet. Certainly, this is our goal. So what is the fundamental problem that with this challenges, I would say, with this technology that's still far from broad impact on biological research, giving us an explicit view of biological processes at arbitrary resolution. One of the problems is very obvious from this movie, that we do this movie and we actually play this movie for about 30 seconds. So the isolation of individual molecule is a strong prior, is also our limitation. You have to have each molecule isolated at different frames. If they overlap, it's difficult to localize. So therefore, you need thousands, and in many cases, millions of these frames to localize one single molecule image. And therefore, this method is slow in terms of temporal resolution. It's difficult to put them into a live cell dynamic imaging. Second, as light as the sole source of molecular positions, they go through different tissue structures. While different tissue constituents will have different speed of light. When light travels through the nucleus, it's slower. When the light travels through the cytoplasm, it's a little bit faster. Therefore, if you have a spherical waveform that propagates through the specimen, you will have distorted waveform, which we call aberration. If you have a lot of waveform distortions, we call it scattering. But basically, if you're trying to do super resolution in tissues, you will face this two dominant effect that does not allow you to get a very high accurate precision of the individual molecules. At the same time, although that we can propose this method potentially localize a molecule with enough resolution, but going towards 1 nanometer to 5 nanometer resolution is extremely important, but still not fully resolved, not fully developed. And this is potentially now being closer with the development of influx localization algorithm or localization methods. However, the localization density is still a fundamental limitation of the resolution achievable in that system. So how can we improve the resolution down to the level of individual, for example, if you aim to resolve the second structure of the proteins, how can we achieve that? These are three different directions in the lab we're trying to focus on. First one, we want to image dynamics instead of fixed structures. Over here showing some preliminary result on that. And we have a classical encoded pits move during the acquisition. It's about 1 minute acquisition. 2 second snapshot, you can see the ring. If you project this into movies, you can see the ring actually move during our acquisition of 1 minute. This is a 3 second snapshot of focal adhesions and actin filaments on fission yeast. And we also want to move super resolution from cover slip surface imaging technologies to tissues and maybe small animals. And here's some preliminary result on imaging amyloid beta fibrils in mouse frontal cortex. We're also developing unconventional imaging technologies or optical instrument to encode more information per photon count. As you can see that fluorophore potentially photo bleach after about 500 to 5,000 photons. You only have limited currencies of your resolution, which are indicated by photon numbers. However, the fluorophore photo bleach, therefore, the key to unlock the higher information content per photon count will drive us towards higher resolution, which has to be encoded first in the physical system before analysis. So what our image looks like, and there's showing 3 different imaging modalities that we have in the lab. And there's the astigmatism. You can see individual molecule blinking on and off. Instead of they are looking at round molecules, they are looking at elongated in one direction or the other. The shape of this molecule actually give you the Z information. You can also look at the biplane setup, which basically imaging the same field of view, but at adjacent axial positions. And therefore you can compare the shape or the width of the different points by function to see where the molecule is. But more interestingly, you have this 4-pi based single molecule imaging where we do 2 objective coherently detect the fluorescence from single molecules, and their fluorescence will be coherently combined together, forming interference pattern. And therefore you can see this pattern is like elongated blob or dots, but they actually some type of like very complex structure like brackets or brackets with 2 dots on the top and bottom. These are individual single molecule fluorescence interference patterns that give you high amount of information, and in this case, in the axial direction. So what we can do with these technologies, for example, we can try to image T7, and these are the phages. These are the viruses of bacteria. When you look at them in super resolution imaging, they look like a cluster of dots. And individually, they're about 50 nanometers, extremely small. And we label the shell structure so that we can see how they shape in their shells. If you tilt around, you can see these images look at little rocks on the cover slip. And indeed, this is a 200 nanometer scale bar. Over here is about 50 nanometer on the axial duration. If you cut cross sections, a lot of them, you can see where they store their DNAs, their shell centers are hollow. And we can merge a lot of them together. For example, here uses template free structure averaging, and you can form a general structure where you can take horizontal and vertical slices, give you pentagon and hexagon shape. You can also do some quantification out of this. You can also image 3 dimensions in whole cells. For example, over here, I'm showing syn-amino complexes. Basically the linkers between chromosomes. And usually how you do this, you have to take a cell and we call it over here spermatocytes. You fix the cells. To image that in one plane, you have to break it such all the chromosomes form on the cover slip surface. Then you do a confocal scan. You see all these 19 strands going over here. And in our system, this is actually coming from the system that Sheng also worked on during the time that she was a postdoc in my lab. And you can see over here, individual strands going at different orientations. We don't have to fix the cell anymore, or we don't have to break the cell anymore. You look at them in their original 3D geometry. Not only that, you can see they are not individual lines, they are actually double lines. These double lines are like roller coaster rails, and they rotate against each other, but they never touch each other. And this is important to highlight that the data that we generate and it's not normal imaging data. There are individual locations of molecules. Each of the location of molecules marked by a block over here basically give you a spreadsheet of all the molecule positions in X, Y, and Z. If you're doing live cell imaging, it will be X, Y, Z, and time. Where you have high density molecules, it will give you higher density. And you can also see other molecules that are not bound to a certain structure where they are in the distribution. And you can have some fun with it. Sheng further improved this method by developing a coherent pupil function, describing the electric field on top and bottom objective to further enhance the resolution from what we just displayed before. Over here, you have a astigmatism-based imaging where you see the elongation of the axial direction because they are usually poor in confocal and these wide field systems that poor axial resolution induce these kind of blurs. Over here, you can see the original contrast-based method. You have significant improvement in resolution due to the coherent detection of single molecules. Shall develop a further model that refine the optical system such that even you get a higher contrast. In fact, that we also demonstrate the possibility of getting very high precision of localization with a very limited number of photons. And these are only collected by 1,000 photons per objective. Okay. And in focus, you already get a precision around 2 nanometers. And even at autofocus regions, you almost cannot tell there's a molecule, your precision about 6.3 nanometers. So how do we do this? The concept is basically locating a molecule based on its shape. It's very, very simple. Over here, we see the astigmatism. That astigmatism has a circular point when it's in focus. When you're going out of focus, you will have elongation in one direction. Out of the other direction, will give you elongation in the other direction. And biplane system, in the middle, they might look the same because they're offset by their axial positions. When they are going out of focus, they all go out of focus at different rates. And therefore, you can see the difference between their points by function. For pi single molecule localization is a little bit more complicated, but let's just focus on one of this line over here. You have a constructive interference in the middle. If you go out of focus, you might have a destructive interference and then constructive and destructive. At the same time, the overall shape of your points by function also modulates at the astigmatism because we put some astigmatism aberration into it. And therefore, the interference pattern over here gives you a huge amount of information gain in the axial direction and also in the XY direction when they're out of focus. So to understand how these data are get, and you first have to get a calibration. So how do we know what shape of molecule, that shape of the points by function is at certain location, right? That's basically our ruler or calibration data set. How do we calibrate? How do we know what type of shape to expect? Therefore, we can localize the molecule center very precisely. We usually do this. We have a bead and put it on the cover slip. And we scan the speed up and down with a very high precision piezo stage, usually about 0.3 nanometer in resolution. We can put this very accurately, okay? And we look at their points by function. In focus, we see a very good points by function that are sharp. Out of focus, it gives you an elongated points by function in one direction, in the X directions. And the other autofocus directions give you the other directions out of focus. Now, what you can do is you can build a calibration matrix, and this is a conventional way of doing it. Sheng and Keys has much better way of doing this, but in general, this is what we do. And over here, you have the sigma X and sigma Y plot. That indicate if you're at around this 1,000 Z equals, we just add the Z number, which is artificial choosing, basically a reference offset of the Z position. At certain locations, the molecule look, the PSF look around, therefore they're sigma X and sigma Y width in the X and Y direction, they're the same. However, for example, you're out of focus away from 800 nanometer on this side, you will have a smaller sigma Y over here, and you have a bigger sigma X over here. That indicates your points by function stretches in this direction, right? Vice versa, you can see over here, your points by function stretches in this direction. So basically, this curve gives you how your points by function going to modulate at different Z position. Now you take your cell and you put your cells into the microscope, you can image that, and the cell give you points by function at different shape. You can then pinpoint, okay, my points by function is stretched in this direction a little bit, in the X direction, stretched in this way. You can find out, okay, what is the Z position? Basically, that's how you do 3D localization. And in this case, it's stable. However, there's a fundamental flaw with this. And this has been done in the single molecule field for a very, very long time. These all calibrations are in vitro because these photons, if you think about this bead going through the cover slip directly, and it doesn't interfere or affected by the tissue. However, when you're trying to do tissue imaging or cell imaging, these single molecules, the photon has to go through the tissue first before being detected by the cover slip, or being detected by the collected by the objective and detected by the camera. And therefore, what we call this is the in situ PSF, where the photons experience sample independent aberration. And really, what is the response after the sample dependent aberration? It will depend on the sample. In many cases, within one sample, it depends on the field of view you're trying to image. If you do not consider that, and you're trying to use a point spread function that you acquire based on bead on cover slip, this is one of the cases if you have significant spherical aberrations. We image mitochondria and within the COL7 cells, and we label the membrane proteins, and we image them through a 94 micron cavity of aqueous solutions. And over here, because of significant spherical aberration going on, your point spread function basically all become wrong. Actually, this is actually a benefit for 2D imaging, but I'm not talking about that. In axial directions, it has very little modulation if you change the Z position of the molecule. Basically, no matter where your molecule is in the Z positions, they all look wrong. And therefore, if you're trying to use a localization algorithm to localize that, you get strap patterns. Why is that? If you just do one optical sections, you will have one strap. If you move your optical section because you want to image a large volume, you get another strap, basically because your round molecules are always assumed and being localized at the center of the axial positions. In fact, that we can quantify this behavior using Kramer lower bound, which gave you the theoretical precision limit and independent of your algorithm development. This basically gives you a theoretical bound of how precise your informations, how precise you can localize your center. And of the in X directions and XY direction, we call it lateral over here. You can see this is not much difference with the increasing depths. Actually, lateral resolution does not suffer. That indicate also agrees with our data previously. You can see in the XY direction, it does not change too much. However, in the axial direction, you can see merely 30 micron into depths, you have significant increase in precision. Increase in precision translates to worse precisions. So basically, you increase the uncertainty of localization. And this is coming from the data credit from Li Fang and showed up in Sheng's annual review of biomedical engineering paper published in 2022. So I'm going to discuss two methods that you can potentially push super resolution despite the significant aberration you're going to see into tissues. And one of them called correcting the aberration. So we can correct the wavefront, just like what usually the astronomers will do. We certainly learn a lot from astronomy in this kind of technologies. And we can do adaptive optics on them. And we will combine some recent development. I will show you how we can do that in a little bit. We also want to retrieve the PSF after the treatment because every compensation, you cannot guarantee your PSF will be coming to the back original PSF. There's no guarantee that. And in the end, you still want to understand what is my point spread function after my correction. And this I'm going to talk about in situ point spread function retrieval. Let's first talk about that. To understand how do we correct aberrations, how these aberrations occurred in the first place. If you have, for example, a GFP next to the, over here, the ER, and they will generate spherical wavefront as they light up the fluorescence. And these spherical wavefront usually are collected by the objective and converted into a point wavefront. And after that, through tube lenses, you form this point spread function that looks very ideal. However, as I mentioned, light propagates through different constituents of cells differently. It travels at different speed. And therefore, this wavefront, after going through different speed materials, will be distorted. In fact, what happens is usually this. You have a distorted wavefront, and you collect a better objective. You have a distorted wavefront instead of a plain wavefront. After that, you can collect better tube lens and then focus on point spread function. And therefore, for example, this is showing an aberrated point spread function over here. It's just very difficult for you to tell where the molecule center is. Right? So instead of fitting a Gaussian, you have to fit something that is more close to the truth. What we can do is with respect to this aberrated wavefront, we can potentially add an element, and we call it deformal mirror. This has been widely used, for example, in astronomy, and we learned this technology from the astronomers. Over here, all these aberrated wavefront will be reflecting based on this curved mirror. If we curve this mirror just to the right angle, then the output of these reflected wavefront will be plane again. And therefore, you can hopefully get this PSF restored to the original manner. Then the question is, how do you control these mirrors? And I'm going to show you a couple of these methods. So we use a deformable mirror, which has 11 by 11 elements across 12 by 12 elements across the board. And these elements can be controlled using voltage differences. And if we can input a voltage map, it will form a different step, a different position of the actuators. And we have reflective membrane on top of it. If you put this into the pupil plane of the objective, you can control the wavefront uniformly across all the field of view. Basically, these distortions that you introduce or compensate will be uniformly across all the field of view due to the geometry of the pupil plane or due to the position of the pupil plane. And you can use this type of mirrors to introduce, for example, aberrations, if that is suitable for your technology or need. Or you can go deeper into the specimen, for example, correcting first and second order spherical aberrations. And here's a just rough demonstration of quick demonstration of how these deformed mirror, if operate correctly, can perform these compensations. This is using a simplex algorithm, trying to optimize the sharpness of the image using a simplex routine. Over here, you have there's a 14.4 microns of depth. You can see already elongated PSF. After simplex AO, you can see the restored sharpness of the metric and also the intensity of this PSF. And over here, you can see 45 micron depths. You have much more elongated. And all the way up to 172 micron depths, you can see really elongated PSF, give you very poor axial resolution if you're trying to image those. But after the adaptive optics, you can see simplex AO really restores the response. Not quite there yet, right? It doesn't restore all the way to the original PSF, but certainly give you a contrast. So to quantify that, we can actually measure the metric. We just implement the shape metric to quantify the shape modulation at different Z positions. So it's basically the width in the X directions divided by the width in Y minus the width in Y direction divided by width in X. If you do this, you will find out, okay, if you have different Z positions, the astigmatism points by function give you a response almost linear. It's not perfect linear, but this is a demonstration of how fast the modulation of these PSFs will give you. If you do an imaging with a little bit deeper into the sample, for example, over here, 12 micron into the specimen, you can see the modulation is significantly reduced. After doing AO, you can rescue some of the modulations. And if you do this, for example, for higher depths, over here, we can focus on the red curve over here, 151 as the imaging depth. And you can see this is hardly any modulation before adaptive optics. That means your point-spread functions stay the same shape throughout very much large depths. That is not very good because the shape change give you the Z information of the localization. So after doing adaptive optics, you can potentially restore this to the manner that you potentially can do localization anymore. However, it's not enough. What we initially do is we further increase the adaptive optics while we further introduce further astigmatisms using the deformable mirror such that the response is almost stationary. And therefore, we can imagine this potentially give you a point-spread function that has the same modulation at different depths. And we use these type of technique to image, for example, mitochondria over here at 96 micron depths. Instead of seeing this over line structures, you actually see a ring that indicates the surface labeling of top 20 proteins. So with that, we can potentially implement this with into the brain and to allow single molecule imaging and super resolution imaging into brain slices. And Michael and Paul Hassenways worked together on this project together. And you can see these are individual fibrils of amyloid beta fibrils that in mouse frontal cortex. And this is for Alzheimer's disease models. And these are fibrils are believed to be one of the causes of these symptoms in Alzheimer's disease. Over here, you can see individual fibrils that can be well resolved, and the crosses between fibrils can be resolved over here. Compared to diffraction limited image, in this case, you have much higher resolution in this case of high because of the super resolution technology, and paired with adaptive optics and diffraction limited image, super resolution, diffraction limit, and super resolution. So I just want to show you another slide. This is not much into depth, so you can see this is fairly shallow, and just on the surface into the brain. It's about 5 micron to 3 micron depths. Over here, you can see this is super resolution image. If you're trying to image that in confocal, you will see you will not be able to resolve the fibril bundle over here. That falsely will give you a biomarker that is not accurate for your diagnosis. So far, I talk about how do we use adaptive optics, and I want to talk to you a little bit more on that. But let me introduce this technology we are actually developed in 2018. That is, we use deep learning to infer the information, or we call it multiplex information carried by a single molecule emission pattern. And we call it SMSN. It stands for Deep Learning Neural Network for Single Molecules. So single molecule blinking data that we take on everyday basis and actually contains a lot of information. And each of these molecules can contain these emission patterns contain information about their positions, wavelengths, dipole orientation, and wavefront shape. And in fact, that it is very difficult for us to write an inference algorithm from this to give you these measurements. For example, if I play this movie and I try to imagine that can I write an algorithm such that I can extract how much coma there is from this movie. And this can be very, very difficult. And aberrations have many, many different modes. For example, 28 different modes of learning keys that are dominant in your specimen. If you're trying to do an inference algorithm or develop an algorithm to do this, to extract all these information, it can be very challenging. So high dimensional information retrieval from these single molecule patterns, although they do contain those informations, are limited by the challenges in regression-based algorithms. So Sheng and Pei actually work on this very hard, and they come up with this solution. So if we want to understand what does the information these molecule carries in terms of 3D location, orientation, and the wavefront distortions, and they can develop a deep learning algorithm. These deep learning algorithms, you can train the network with simulated or experimental data set. After training, you can give them these very complex patterns. The deep learning is still going to work and give you some emission, give you some estimate. So they not only demonstrate it's possible to do inference, it's actually their network is designed with very unique considerations. They train their network with information-based cost function. Basically give you how much statistical information for each of the training data set, and you weight that in their cost function so that the training does not force the network to do something statistically impossible. I'm not showing that over there due to time limit, but I want to mention that this network does not only does inference, it actually extracts the information at the statistical limit because we can localize single molecules all the way to the lower bound. But I want to show one of the demonstrations that is very interesting and lead to the next project I'm going to show you. Over here, we just let the network to monitor individual molecule patterns. And you can imagine these emission patterns will change based on their aberrations. And we can roughly tell as a microscopist, I look at the image, I can tell, yeah, well, you have some coma over here, or you said some spherical aberration. Why don't you change that? But why don't we let the deep neural network do that and tell us what kind of aberration there is? And they can tell much better accuracy with much better modes, much more mode that they can estimate. So over here is a test, and we have single molecule blinking events where Sheng and Pei actually in the room and clicking and generating arbitrary aberrations and didn't tell the network. And we'll see whether the network can actually infer it back. So over here is a real-time updates on network inference. And over here is a network estimated pupils, and we can start moving. In the beginning, nothing happened. Now, you can see they generate some astigmatism, and the network gave you the response of that. So the deep learning network actually goes through the system, look at that, and understand the system such that it give you, yeah, well, currently you're giving some diagnostic symptom. Now you change it back. You can see this response is almost in real time. So this is a very, very good tool and stunning for us, actually. When we get into this project, we didn't even think deep learning can do this, but this actually is available. So what we can imagine this, deep learning can potentially drive the deform mirror in real time to do something, to do real-time compensation on the fly. So Pei Yi worked on this project after we submit that paper, and I think it's already a very interesting paper. We submit that, and that got accepted, published. And Pei Yi worked on the follow-up of this project, trying to basically develop this feedback loop. Okay, it turns out to be the project is much more difficult than we thought. So it's very different when you sit on the passenger seat and estimate what's going on around the car, than you sit in the driver's seat and trying to drive the car. The difference is that whether you can estimate or whether you can control the instrument to actually do proper things. So over here, Pei Yi developed this. Over here, we have a general flow chart of this. We have raw data from single molecule blinking result. You have these very aberrated point spread functions. We go through SMNet, which we revalidated and multiple round. This is still a very good network to use. And we put that into a common filter because every time you drive the instrument may not respond perfectly. For example, you turn the car wheel a little bit, it might be turned a certain distance, but due to maybe you have slippery, due to some other conditions, you have uncertainties in your control. The common filter actually is very good at that, taking both the previous history of estimations and current predictions and choose the proper manner. And after that, the prediction will go into experimental mirror mode. This gives us understanding what type of aberration, how much, and these are numbers you can see over here. We translate that into mirror actuator voltage maps and then drive that to correct aberration. So over here, you can see some demonstration in steel graph over 0.75 and radiance in wavefront errors. You can do adaptive optics, you can do this, and DLL represent deep learning driven adaptive optics. And over here, you have stochastic aberrations over here, but after the compensation, you restore the sharpness of this image. And this is even more aberrations, you can restore the performance of them. So here is another movie showing how fast this aberration can be compensated. And over here, I'm gonna play this movie while the deep learning basically look at this movie and trying to guess what's the aberration and control the system. Step by step, you will see a phrase point, that means the system updated. Very bad point spread functions. With one shot, the deep learning actually can drive your system back into normal and give you very high contrast images. And this continuous movie basically continues, and we can do this for the rest of the for the rest of the iteration, it will not diverge. We give some more interesting tasks. So now we have some aberration in the system. We let the deep neural network to correct the aberrations, drive the deformable mirror. And then we randomly give some random aberrations without telling the deformed mirror when we give it to him and how much. Basically, it's a blind test, which is introduced at arbitrary times. Now let's play this. Over here, you can see the aberration goes down because it's compensated. And then you have some aberration over here. You can see this wavefront distortion goes down immediately. This is in play in real time. You can see how fast this aberration can be corrected by the deep learning driven adaptive optics. And this can continue on for many, many cycles. This entire movie is only 3 minutes, but we introduce more than 10 types of different aberrations. And without miss, this one correct them every single time. With that, we can potentially image biological structures at significant depths. Over here, it shows dendritic spine image where you can see dendrite in the middle. You also have these tree-like structures and shows the spines. And this is diffraction limited image. This is super resolution image. You can see the drastic improvement in resolution. You can actually identify a lot of spines. You can quantify the spine volumes due to the high resolution nature. You can also much better distinguish their spine types because before you have this image, it's very difficult and really easy to mislocalize or misidentify. All of that is actually be done in 134 micron depths, which is significant increase than previously whatever I showed you, about 20 to 30 micron. We have many of these movies and videos that you're welcome to check that. This is in a preprint server right now. We can quantify the neck and the head with very high precisions. And in addition, we can auto quantify the volumes. And this has been important markers that we're trying to diagnose whether behavior changes in mouse actually has correlations with these span volumes. Let's now change topic to another work. And even though you correct all these aberrations, right? But there's cases that your system may not correct enough such that you have some residual aberrations. Or let's say you're in a system that you do not know how to use adaptive optics and still want to do tissue imaging. So this is the method that we developed for in situ point by function retrieval. And this is developed by Fan and Donghan, who already got their faculty position in China. And they basically developed this algorithm that allows you to retrieve an aberrated points by function directly from single molecule data set. And you can see the amyloid beta fibril structure over here. This is in diffraction limit. And if you do that in super resolution image using InSpar, you can see well-resolved structure and individual filaments can be traced in three-dimensional manner. Let's see how it was done. The problem is, as I mentioned, you have a single molecule over here, give you the calibration data set. The calibration data set is not accurate. So whenever you go to tissues, you do not know what's going on with your points by function. So how do we solve that? Let's ask the question in a different manner. PSF calibration using B is in vitro, and that is not accurate. So accurate single molecule localization requires in situ PSF where the molecule is, and especially in sick specimens. So the question is, how can we obtain an in situ PSF? If you look at the points by function, or if you look at the data we have, we're certainly not lack of points by functions. Actually, all we take is points by functions, right? Single molecule blinking images. Every blink is a points by function. If you segment that into different optical, different subregions, you can get a collection of points by function. And these collections should come from the random sampling of different molecules at different locations in the Z dimension. So basically, you do not have a points by function, but you have random sampled images where you do not know where it's sampled from. So the problem is that when you take these random data set, you do not know where they are sampled from. If you know this, you just concatenate them together and form a in situ points by function. If only we can assign each emission pattern to its correct position. And InSpar actually just did that. With single molecule blinking data, we segment all of these single molecules at different locations, form a PSF library. And after that, we can use the initial guess. This can be just a flat pupil or some theoretical astigmatism pupil. Over here is a theoretical pupil we put in there just to give the general orientation of the astigmatism shape. And after that, using these reference PSF, we form a template and then assign by similarity of each of the PSF into different bin in the axial position. So similar looking PSF will locate it here. Another simulated PSF, a group of another similar PSF will locate it at, for example, plus 800 nanometers. And this gives you nonetheless inaccurate binnings. However, it's a first step. What you can do is you can average them, rotate them, and sorry, not rotate, but you can average them, align them, average them, or sum them to give you an updated pupil, which represents the electric field in the pupil plane. And after that, you can generate an updated reference PSF. This certainly is a little bit closer to the PSF library. And then you can do this assignment again. And then you do this over and over about 7 times or 6 times in our experience. You can get this in situ PSF. And this PSF, for the first time, makes single molecule localization in cells and tissue, potentially represent accurate points by function. So we can use this to construct an in situ 3D response directly from single molecule data set. We can also use this data, PSF to pinpoint their locations with high precision in whole cells and tissues. And here's some demonstration using simulated data set. Over here, you can see two plane images of randomly located point spread functions. If you use the algorithm, you find out the pupil function in situ and ground truth. You will find out that the difference between these are very small. In fact, the residual is showing over here. If you plot the Zernike coefficient or amplitude of these, and you can see they are very, very close together, with the ground truth being the circle. The diamond, a blue diamond, is in spar. You can see in each different mode, they're extremely close. Also over here, demonstrated retrieve the PSF and the ground truth PSF. You can see they're very accurate. We can also do some tests also using deformed mirror. What we can do over here is we have a blinking molecule over here, and we can change the deformed mirror. And by changing the deformed mirror, we also look at the blinking molecule to let the InSpar retrieve the aberrations to see whether it can retrieve accurately. And over here, this is InSpar retrieve the amplitude. This is DM input. This is what we expect, but this is what you will retrieve. You can see they are mostly diagonal, and there's some tertiary aberrations we're having a trouble with, but in most of these cases, they're fairly diagonal. With that, you can look at, for example, image whole cells, and knowing that you can potentially, you can get an accurate point spread function in comparison to whatever is the in vitro calibration based on B. And this is the InSpar. You can see the sharpness increases because you have a higher accuracy of point spread function that directly extract from your data set. Over here, here's the InSpar reconstruction. Here's the in vitro reconstruction. You can see the difference. And over here, you can see the difference between point spread functions. So over here, you can see in vitro PSF is apparently much better than the InSpar PSF because InSpar shows spherical aberrations. Basically, your system has spherical aberration. InSpar directly look at the data and extract these. And the difference between in vitro pupil over here and the InSpar pupil shows why there's a difference between these reconstructions. And with that, you can image, for example, mitochondria. This is actually deep into the specimen. And we labeled 20 proteins that are on the membrane of these mitochondria surfaces. And ideally, they form a very nice layer of enclosure of the mitochondria structures. And to visualize that, we can potentially cut cross sections. And this is 200 nanometer thick running cross sections. You can open up the mitochondria and close it as you're running through it. And the total localization over here is 1.1 million, and XY precision is 8 nanometers, Z precision is about 21 nanometers. So we also have a reconstruction of amyloid beta fibrils. They are Alzheimer's disease models. And over here, you see that same rendering. You can see the individual filaments running through the three-dimensional space. Instead of a cluster or size of the plaque, we can now quantify their three-dimensional orientations and their densities. And all of that can be important biomarkers for potential diagnosis. And if you do cross sections, you can see the fibrils running up and down in its three-dimensional volume. All right, let's move on. So I want to thank all the students and postdoc who actually did the work. And you actually can see Mike Wisnowski over here developed the simplex adaptive optics. And Sheng is showing over here, her work is instrumental in many of our publications, including our full PI and annual review, and that one has coherent pupil functions. And also other lab members who contribute to this work and our collaborators. And without stopping, I want to show you a couple of pictures from the lab. And these are different pictures at different years. And for example, this is very early on in 2016, we showed a picture. And these are during COVID. Not only you can see our lab members over here, you can see our visitors, Professor W. Murner, Martin Booth, and very talented soccer player, Yaya from Ivory Coast. So the lab did different things. We are developing new and unconventional optical instruments. So you can see Mayam over here, Lee and Haozheng is showing their instrument. And we also have organic chemists developing probes because single molecule probe is very important so that it is bright and also switches. And we also have collaboration with IU School of Medicine, whereas Medi is an MD PhD student trying to handle mouse there over there. We have strong interest in algorithm, statistics, and math. And over here you see Elun and Vemera developing algorithms. Cheng is our cell biologist who works on cytokinesis and look at fishing yeast. And some of his work I didn't show today. We're also very much into artificial intelligence driven adaptive optics and imaging in general. And hopefully that will make a difference that free many of our imaging sessions and with automatic manner. So with that, I would like to thank you for your attention.
Questions in the back?
Thanks for a great talk. My question is, it seems like most of the imaging that we've done so far is for small animals like mice and stuff. It's really neat, but I'm wondering whether the goal is to move towards like human tissue structures and stuff like that. And I'm wondering whether there's a path towards that as well.
Yeah, actually, this is something we actively work on. And one of the brains that we initially want to image is post-mortem brains from Alzheimer's disease patients. And these brains actually have a different fixation protocol or embedding protocol than usually the brains are. So protocols in how to treat these post-mortem brains to reserve their structure at the same time minimize fluorescence background, which in single molecule imaging is very, very important that you do not want your background to overshine your single molecules. That is a part that we're still working on. But yes, that is possible. But in terms of live human imaging, that is very difficult at this stage because you have to label your tissues and with fluorescent markers. I would say Raman imaging, for example, these label-free type of imaging technology will be more suitable towards non-postmortem, for example, tissues. Yes.
So I have a pretty shallow knowledge of adaptive optics, a very shallow knowledge, but I am curious about the deformable mirror and sort of like the scope of wavefront errors that you're correcting for with it. So are there kind of like material limitations on the wavefronts that you can collect for or correct for based on like the deformability of the mirror? Like, do you ever run into any issues where like the actuators that are like neighboring each other are pointing in two very different directions to be able to correct it, and that causes complications?
Yeah, that's actually one of the reasons we use common filter. So as you can imagine, the coupling between the two different modes, two different actuators can have significant effect on this accuracy of representing of the wavefront. Yes, that is a big issue. That is something that has been addressed multiple rounds with different approach. None of these approaches are very straightforward. And it requires a lot of modeling and tests. Basically, you can try to generate a response function by measuring using a shock harmless sensor or interferometer. And you can see, okay, at different voltage and what type of shape there is. It's not linear. And therefore you have to do a lookup table of these actuator mappings, which we didn't implement over here. And we found that the common filter already is good enough, not only accounting for that inaccuracy, but also in the inaccuracies when you have a lot of single molecules that are coming from with different intensities, and therefore their measurement uncertainty are different at different point. And therefore we use that as a combination in common filter. But that is a very important question. I think it's still to be solved. Yep.
Thank you very much.
Thank you so much for your nice talk. So I'm wondering for your 4x microscopy technique, how do you use some direction having as to occur, and also how many detection direction that we use to correct your fluorescence line? Also use that setup, how many camera do you have to introduce to image from all of the 360-degree correction?
Very, very good question. So I will say we are doing the bare minimum. We just use tool. and each of the objective is 1.4. There have been theoretical studies probably proposing 3 objective detection. There's some improvement you can potentially do because you cover more spaces. But the problem is that your objective is bulky. Putting 3 objective, that requires using high working distance objective. And therefore your NA will be smaller. So 2 directions, right? And then I didn't show that, but please come back if you were interested. And you can collect that by combine that by 50-50 beam circuit. And before that, you have SP polarization of birefringent material such that you introduce SNP with different phase delays. In the end, you will have 4 different images, top SNP, bottom SNP, and they will have 4 different phase delay. And then you collect them into triangle mirrors and put them into the camera. So just use 1 camera. You can collect 4 different quadrants, basically measuring 4 different phases of the interference pattern, such that you know exactly where you are in the interference phase.
Thank you. So if you give us any optical diagram that you have already published, I can do it.
Oh, definitely. Yeah, I can show it. Yes.
Yeah, back there.
Great talk, first of all, that was really cool. I was wondering, and this might be a little bit ignorant about like gene click, can you apply some of the asymmetric mirror or the deformable mirror to a light microscope and maybe correct some of the working through the mirror as you pass through the example that way?
I think that's maybe well suitable to do that. But in light sheet, I would suggest SLM, because in our system, we use deformable mirror because it's not polarization dependent. SLM in your light sheet, you can just throw half of the light and you still get very high controllability because the pixel-based SLM is much better control of the beam quality. But yes, you can potentially generate stapling on there. You can potentially pivot your beam and correct aberrations if you want. Yes, very good question. Yes, please.
On the correct marker of the aberration.
No, we correct 28 different modes, all the way down to 2nd order spherical. And tertiary mode, we don't correct. Yeah, fairly. I don't think there's a limit for us to correct. And Pei even tried the 55 modes and it works. So, but in our opinion, that the aberration generated by these steps of specimen is not that complex. So 28 mode is good enough for us to start.
All right, let's stop there. Thank Fang Huang. You can show it, but you were.
They know why they can choose.
Wait.